So, life itself is regulated by catalysts. Most catalysts are enzymes, and they're mostly made of protein, although it's been recently discovered that some of them are RNA. Basically, enzymes allow reactions to happen much more quickly. Imagine this. You have a friend that lives on the East Coast and a friend that lives on the West Coast, and you think that they should both be together. Now, over time, they could potentially end up in the same place. However, it might take a while for them to end up in the same place and meet. So you, as the enzyme or the catalyst for their relationship, call your friend on the East Coast and you call your friend on the West Coast and you say, why don't you come out here and meet me in Colorado? And they both come out and meet in Colorado and fall in love and blah, blah, blah. You were the catalyst for their relationship. They may have run into each other at some point in time, but it happened much more quickly because you saw the similarities and you drew them together. Enzymes basically function in this way. Enzymes draw things together. That is, they stabilize a temporary association between the substrates and then let them interact. Enzymes are not changed or consumed in a reaction. So in an example of matchmaking, nothing happens to you as the catalyst after bringing your two friends together. You still exist and you have the potential to bring another two individuals together. Now let's look at a biological example. Carbonic anhydrase is an enzyme that allows for transport of carbon dioxide through the blood. It takes water and carbon dioxide and brings them together to form carbonic acid. We looked at this earlier in the semester when we considered acid bases and a buffering system. This again is a buffering system. And you may recall it looks like this. We have CO2 plus H2O, and it yields carbonic acid. This reaction can proceed in either direction. Now, if there were no enzyme present, you could get about 200 molecules of carbonic acid per hour made within a cell. But when we add the enzyme carbonic anhydrase to facilitate this reaction and bring our carbon dioxide and our water together, then we could get 600,000 molecules per second within the cell. That's a pretty marked increase. So the bottom line is that enzymes speed up reactions by drawing the components together and facilitating the reaction between them to form the products. Enzymes, again, they're not changed or consumed in the reaction. So the carbonic anhydrase is an enzyme that's a protein, and it can continue to pick up CO2 and H2O and turn it into carbonic acid. Enzymes, in order to do this, have to have an active site. That is, they have pockets or clefts for the binding of substrates. You can see here is the active site in this enzyme. The substrate, or the molecule that's going to be changed, moves into the active site, and then the change occurs, and the enzyme releases the substrate. We often view this enzyme-substrate interaction in this sort of equation form, where we have an enzyme plus a substrate leads to an enzyme-substrate complex, which then disassociates into a product, leaving the enzyme whole and able to do more work. And this is exactly what we see figured down here. We have an enzyme with active sites for two particular molecules. The molecule sits in the active site. Here we're hydrolyzing the bond between these two sugars. So we add water and we're able to split the two molecules apart. We now have a product of glucose and fructose, and the enzyme remains, so it can go and pick up another disaccharide. Basically, the enzyme applies stress to distort a particular bond to lower the activation energy. Enzymes are said to have an induced fit. That is, that once the substrate joins with the enzyme basically grabs onto it to form the enzyme-substrate complex. Each enzyme is very specific, that is, it has a precise fit to the substrate into its active site. So the active site is just the right shape and size to fit exactly what it is the enzyme is supposed to act on. So there are hundreds of thousands of different enzymes that are involved in all of the metabolic processes in our bodies. Enzymes can be suspended in the cytoplasm, or they could be attached to cell membranes and organelles. That is, they could be free-floating, or they could be attached to things. Sometimes we'll see multi-enzyme complexes, that is, that different subunits work together to form a molecular machine. That means that one complex of enzymes in one location is able to do a whole series of reactions. The product can be delivered in this way easily to the next enzyme in the complex, and the product can be formed in the same location. Because of the close proximity of all these different enzymes, we can avoid having unwanted side reactions. All the reactions can be controlled right there in the multi-enzyme complex. Until recently, it was considered that all enzymes were proteins. However, in 1981, Tom Cech from the University of Colorado in Boulder discovered that certain reactions are actually catalyzed by RNA molecules, and he called these ribozymes. There are two kinds of ribozymes. There are those that are involved in intramolecular catalysis, which is those that catalyze reactions on RNA molecules themselves. Or there are intermolecular catalysis mechanisms where RNA acts on another molecule. More recently, it's been discovered that the RNA in ribosomes is not just structural, it actually has an action. So that means ribosomes themselves are ribozymes, enzymes that are made of RNA. Now, the rate of any enzyme catalyzed reaction depends on the concentration of the substrate and the enzyme. Again, imagine the tennis ball example, just like receptors in the cell's surface. The more catchers there are for the tennis balls, the more substrate or tennis balls that I can move. If there are less enzymes, then we can move less substrate. If there are more enzymes, then we could move more substrate. Any chemical or physical condition that affects the enzyme's three-dimensional shape can also change its rate. So optimum temperature will allow reactions to occur at the fastest rate possible. Now, when the temperature gets too high, the proteins become denatured, as we've learned in a previous chapter. When the proteins are denatured, the active site no longer fits the substrate, and the enzyme can no longer do its work. In this figure, you can see that the optimum temperature for human enzymes is about 42 or 43 degrees centigrade. So that's our regular body temperature, 98 degrees Fahrenheit. The optimum temperature for some of the enzymes in hot springs prokaryotes is much higher. So the enzymes have evolved to accelerate reactions in the particular environment that they exist. Another concern would be the pH. At the optimum pH, enzymes operate at their highest level. However, with a pH too low or too high, the enzyme activity is changed. From this, you could probably understand why a very small change in temperature or pH within our own bodies can really mess things up. Now, let's look at some other things that affect enzyme activity. There can be inhibitors, that is, things that get in the way of the enzyme doing its job. So an inhibitor is any substance that binds to the enzyme and decreases its activity. There are two types. There can be competitive inhibitors that compete with the substrate for its active site, or we can see non-competitive inhibitors. Let's look first at our competitive inhibitors. In this example on the right, you can see that the substrate should fit nicely into this lock and key mechanism or induced fit mechanism that the enzyme exhibits. However, there's another molecule that also could bind to this active site. If the active site is filled with this other molecule, the substrate itself can't get into the active site. So this other molecule is the competitive inhibitor. So, competitive inhibitors interfere with the active site of the enzymes, so substrates cannot bind in that location. Non-competitive inhibitors are also known as allosteric inhibitors. they cause the enzyme to change shape. The key here is that they bind to somewhere other than the active site. So as you can see here, the inhibitor binds to a site that is not the active site. We call this site the allosteric site, hence the name allosteric inhibition. When the inhibitor binds to this allosteric site, it causes a shape change in the enzyme so that the active site that you see here no longer has the same fit that's necessary to accommodate the substrate. And if it can no longer accommodate the substrate, then the enzyme cannot function anymore. So again, two types of inhibitors: competitive inhibitors that compete for the active site, or non-competitive inhibitors, ones that bind to another site and cause a conformational change so that the enzyme can no longer pick up the substrate. We call these allosteric inhibitors. A couple of other factors that can influence an enzyme's activity are cofactors and coenzymes. Cofactors are molecules that assist an enzyme in its function. They're often metal ions like zinc found in the active site of the enzyme. In the enzyme carboxypeptidase, we find zinc. Zinc has a charge on it and it attracts the electrons, and electrons are pulled away from the substrate so that they can sit in the active site. Cofactors are required in the diet in small amounts. Again, they're metal ions. As compared to coenzymes, coenzymes are still cofactors, but they're organic molecules that are non-protein. So coenzymes are non-protein organic molecules. Most of them are vitamins that are essential in our diets, many of our B vitamins. And often coenzymes, as well as cofactors, are involved in capturing and transferring electrons. So they pull electrons into the active site and hold on to them, releasing the molecule, and then transferring those electrons to the next molecule that comes into the active site. That's biochemistry, really, but we'll see some of the examples of that as we examine cellular respiration and photosynthesis. We won't look at it in much detail, but recognize that it's about transferring electrons. The key is transfer of electrons and transfer of energy. We'll see this in the next couple of chapters in much detail. Let's look a little bit more at these allosteric enzymes. They have active and inactive forms. Most of them have non-competitive inhibitors that bind to an allosteric site, as we previously discussed. That's basically like an on and off switch. When an inhibitor is bound to the allosteric site, it could turn the enzyme off. However, it could bind to the enzyme and have a conformational change that opens the active site. These figures below will help you understand this. Here we have a non-competitive inhibitor. The non-competitive inhibitor will bind to this site that's not the active site. Here's the active site. And it causes the enzyme to change in conformation. This change makes it so that the active site is not available for the substrate. So this would be an allosteric inhibitor. However, we could have an allosteric activator. Let's say the enzyme has this conformation normally without the green guy there. And when the green guy binds, it opens up to reveal the active site. This would be an allosteric activator. So that is, it binds to the allosteric site and increases the enzyme's activity by having a conformation change that exposes the active site. So remember, the allosteric site is simply a site that's not the active site. When an inhibitor binds, then the site will close and be unavailable for the substrate binding. However, an allosteric activator could bind and cause the enzyme to open up and allow the active site to be exposed so it can pick up its substrate and thus have increased activity. So we frequently speak of metabolism. What is metabolism? Well, metabolism is the total of all the chemical reactions that are carried out by an organism. So every single function that we have internally is our metabolism. So anabolic reactions, or anabolism, is where we expend energy to build up molecules. I remember anabolism because of anabolic steroids. Anabolic steroids are taken by bodybuilders to build extra muscle tissue. Catabolic reactions, or catabolism, are those reactions that harvest energy by breaking down molecules. So plants perform anabolic reactions in order to capture carbon using the energy of the sun to build organic molecules. Whereas cellular respiration is a catabolic reaction where we break down the food that we consume in order to produce ATP and fuel all the activities of our bodies. So metabolism is built of a series of biochemical pathways. These are reactions that occur in a sequence. So the product of one reaction becomes the substrate for the next. It takes many steps for any pathway to occur, where we have an initial substrate that's turned into an intermediate by one enzyme and turned into another intermediate by another enzyme, so on and so forth, all the way down the line throughout the biochemical pathway. So how is the rate of a biochemical pathway controlled? Often, an end product of that pathway will bind to an allosteric site on the first enzyme in the pathway. When it binds to this, there's a conformational change so that the active site no longer fits the substrate, and it shuts down the pathway so that raw materials and energy are not wasted. Why not intervene at the first step in the pathway rather than later on down the line? This is called feedback inhibition. The end product feeds back on the first step in the pathway to inhibit further action of that biochemical pathway and thus shut down production of the product that we need. So let's take a quick card quiz. Small organic molecules that assist in enzymatic functions are: feedback inhibition occurs when the final product acts as an inhibitor to the first enzyme in the pathway. This is true or this is false? Carbon monoxide binds to the hemoglobin protein at the oxygen binding site. Once the carbon monoxide binds, the oxygen can no longer be transported. What sort of situation does this describe? Is it competitive or non-competitive or allosteric or feedback? Increase in the temperature increases the rate of an enzyme catalyzed reaction. Once a critical temperature is reached, however, the reaction stops. Why does this happen? Here are your answers. Check in with yourself. Be honest. Did you miss any of these? If you did, please go back and investigate. As I said before, this chapter is really important for understanding subsequent chapters. So if you're missing something or you don't understand something, I invite you, please, please, please reach out, contact me, and ask for help, ask for clarification. Post to the discussion board, make a phone call, write me an email.